All right, good afternoon, ladies and gentlemen. Let's get started. So, a couple of housekeeping notes before we dive into the heart. Next Tuesdays and Thursdays lecture will be online only. Okay, so they will be video recorded for you ahead of time and posted online. So feel free to sleep in on next Tuesday and Thursday. Okay. My office hours are also a little skewed next week. So if you need to come and see me, if you could just send me an email and we'll fit you in somewhere, okay? Did everybody hear that? Because you can't talk in here at the same time. Okay, is everybody clear? Tuesdays and Thursdays will be lecture captured for you ahead of time and put online. And then Friday next week, we have our practice problem solving session, so that'll be lots of fun. And then email me if you need office hours for next week, okay? Any questions about that? Okay, let's dive in then. Let's think about where we were. We were doing a general overview of the cardiovascular system, closed circuit system, pumps, pipes. Now and of course, remember, we've got to keep our eye on the prize. Why were we thinking about those things? Because these pumps and these pipes are going to be tools that we're going to use in order to regulate mean arterial pressure. Okay, so we're going to keep our eye on that mean arterial pressure prize the whole time. But just a reminder as to that's why we were talking about this in the first place. Okay, so now we have to look at how the heart and how the blood vessels work and who controls them. Right, so then we know how we're actually going to tap into changing these tools or using these tools when mean arterial pressure changes. Okay, so that's where we're headed. So we're going to start with the hearts first. So essentially, we're talking about, I'm sure you've seen pictures like this before. We're going to start our discussion today talking about the pumps. I have no idea what I was planning on writing there, so we'll try again. I think it was pups. We're not going to talk about puppies today. The pumps, so essentially, we're going to be thinking about the heart. Two independent pumps beating simultaneously, okay? And we'll put together today how that happens. So, two independent pumps beating simultaneously. Okay, so we're just going to basically quickly whip through the flow through the heart today, just as a reminder, just get some little functional anatomy behind our belt here, and then we'll come back and review the valves a little bit later when the valves become important for us to think about. Okay, so just real quick, like we start with blood returning to the heart through. So let's start with blood return. So let's say we're in the venous side. Okay, so we're on that systemic venous side, heading back to the right heart. Okay, so we have flow back to the right heart through the inferior and the superior vena cava. So these are really big veins. Okay, they are two inputs to the right atrium. Okay, so we're going to go here first. Flow into the right atrium. We fill the right atrium, then get flow through a valve into the right ventricle. We need to get that right ventricle to contract in order to eject blood out into the pulmonary artery. Okay, so we're heading from our right ventricle up into the pulmonary arteries. See, so we go here next. Okay, right atrium, right ventricle, out into there's left and right pulmonary arteries. Okay. We then, there's left pulmonary arteries tucked in behind. Here they are. I guess this would be C as well, heading out into the left and the right pulmonary arteries. Okay? So we're heading out into the left and the right lung here. So we're up in that pulmonary circulation now, right? We move through the pulmonary arteries, that pulmonary capillary bed. So this blood that was in here has now visited the lung and is now coming back to the pulmonary veins and coming back to the left heart. Okay, so all we're doing is going around that circulation system that we laid out on Thursday. Okay, so we're coming back. What am I at? I'm at C. So we're coming back through pulmonary veins now. There'll be other right pulmonary veins, but I think that they're tucked in the back of this picture. So they're back there somewhere. I just can't see them. So we're coming back from the left and the right pulmonary veins, emptying into the right atria, or the left atria, which gets no respect on this picture. It's not even on here. So let's go right here to the left atrium. This guy right here. Okay, A, B, C, D, E. Okay, headed back to the left atrium. Left atrium fills. We then fill the left ventricle. Okay, we got to convince that left ventricle to contract. Where's my left ventricle? F. Left ventricle to contract in order then to eject blood out into the systemic arteries. The first biggest systemic artery we have is the aorta. Okay, so we're taking that blood from the left ventricle, headed out through a valve right here, out into the aorta. So now let's go G here, okay? So that's our path of flow. It does happen to be up in this aorta where we have sensors for mean arterial pressure, okay? So these sensors, some of these sensors that we're actually going to build into our feedback system are sitting right outside monitoring the highest pressure our body has to offer, okay? So out into the aorta, then we bifurcate and diverge this vascular tree into those 600 vascular beds, okay? So we're going to go from this aorta to many arteries, to those resistance arterioles, into 600 capillary beds, back to the venous side, and we whip it around again, okay? So general flow through the, general flow of blood through the heart. Like I said, we will come back and visit the valves that separate each of these compartments when the valves become important for us to think about. Okay, but right now, so we've got blood flow through the heart. Okay, now there are two cell types in this heart that are actually going to turn this thing into a pump, right? Because essentially we have to take this piece of tissue, turn it into a pump. All right, so two cell types in here that are going to contribute to making a pump. Two cell types in the heart. We've got nodal cells, okay, and these are going to be our electrical system. Okay. Oh my God. Hang on. All right. So it wasn't recording. I'm just going to quickly put this back up and say, for the video, know the blood flow through the heart. Boom. And we're on to two nodal cells in the heart. Yeah. So the video captured today will not be entirely complete. I apologize. Yes, technical errors. Anyways, know the blood flow. We've just been spending the last, I'm going to say 4 minutes on just looking at blood flow through the heart. And now we're looking at the two cell types in the heart that make this thing a pump. At least maybe they heard me. Yes, they did. Okay. And by they, I mean, I guess you, I don't know. So nodal cells, that's that part of this electrical system. Okay, so we got to excite the tissue, and then the tissue we're going to excite are the muscle cells, the contractile tissue, the contractile system. Okay, so let's deal with nodal cells first, and then we'll look at the contractile cells, and then we'll put them all together. Okay, so nodal cells are cells that can generate their own action potentials, and so you might have run into these in theory in human phys 1. We call them spontaneous depolarizers. So these don't need a stimulus to change their membrane potential. They're going to spontaneously change their membrane potential on their own, okay? So we've got some spontaneous depolarizers up here. So because they spontaneously depolarize, they are then the trigger for they're generating the first action potential, which is then going to be sent through the electrical system of the heart to change the membrane potential of the contractile cells to get them to contract. Okay, that's essentially what we're doing here. And we're going to find clusters of these nodal cells in the sinoatrial node and in what's called the atrioventricular node. So back here, if we just look at here real quick, the sinoatrial node is going to be up here, hanging out in the right atrium, and we'll revisit this in a minute. And then the AV node it's not in the pulmonary artery, but it's kind of tucked right in that pulmonary artery, and it's at the junction between the atria and the ventricles. Okay, so that's where these spontaneous depolarizers live, all right? So let's think about then how nodal cells spontaneously depolarize. So you had previously gone into depth into looking at the action potential of a skeletal muscle cell and a nerve, I think, in human phys 1. And you always started with this membrane potential. You talk about a resting membrane potential, right? You learned how to calculate it. And in a nerve or in skeletal muscle, it's just sitting there constant around -70 or -90 until a stimulus comes along, right? And it will sit there until a stimulus comes along. And once you change the membrane potential a little bit, you opened voltage-gated sodium channels, you got massive flux of sodium into the cell, depolarized that cell, okay? So just trying to land you on something that you might remember. But the issue there was is you had this resting membrane potential, and something else had to come in and say, all right, we're going to change your membrane potential to -65, which is threshold for the voltage-gated sodium channels, and then you had the action potential occur all or nothing, right? Voltage-gated sodium channels open, voltage-gated potassium channels open, depolarize, repolarize, we're done, okay? So to make a spontaneous depolarizer, all you need is to design a cell that doesn't have a stable membrane potential, right? So if it can slowly creep up to threshold, it'll go all on its own, right? So a spontaneous depolarizer isn't some weird thing now that we have to try to invent. All we have to do is say, let's alter a sodium channel and just make it slightly leakier than normal. And now sodium slowly leaks in, the membrane potential will go up, and eventually, if we decorate the cell right, hit voltage to some channel, get it to open, and our action potential goes. We depolarize, we repolarize, and then we're back down. And then we slowly leak sodium again until we hit threshold again, boom, we're gone. Okay, so a spontaneous depolarizer isn't anything magic. It's just a slightly leakier cell. Okay, therefore it doesn't have a stable membrane, resting membrane potential. Okay, so that's what you're going to see here. So this is our action potential for a nodal cell. Okay, in particular, this is the SA node, but the AV node looks very similar. Okay, so we have let's say our lowest membrane potential being around between -60 and -70, or I guess it's probably around -70 at its lowest point, but it's not rock solid there, okay? That we have this slow sodium leak, so we have some channel in the membrane that allows sodium to move, and that's very different than skeletal muscle cell or the nerve, right? We didn't have this leak happen there. So we've got some slow sodium leak. And so we've got some sodium flux in at all times. Okay. There is a channel responsible for this, and it creates this sodium current. It's sometimes you'll see it written as IF. This current And the F stands for funny, because when it was first identified, someone said, oh, that's a funny little current, and there it stood. Okay, so we have this funny little sodium current at rest, okay? But all it really is we've just got a channel in there that allows sodium to move. It's a little leakier to sodium, okay? So we get this slow leak of sodium in then, which means the membrane potential will slowly depolarize, okay? Then this nodal cell happens to be decorated with voltage-gated calcium channels, right, which have a threshold that's hanging out around 40, around this region here. Okay, so eventually we are going to depolarize enough to hit threshold for voltage-gated calcium channels. Did I say sodium before? I meant calcium. Okay, voltage-gated calcium channels. So does anyone remember enough about ion fluxes to know what's going to happen if you make a membrane instantaneously permeable to calcium? Do you remember where the ions, like, was there a high amount of calcium on the inside or the outside? What was the E ion? What was the, yeah. It's higher on the outside than on the inside. Perfect, massive concentration gradient, right? Does anybody remember the E ion for calcium? So when will calcium stop moving if we let it? What would the membrane potential be? Do you remember that? That's a nitpicky level of detail, that's for sure. It was so it was positive, I'll give you that. But it was the most positive we've ever had. Sorry, say that again? So that might be the concentration number you're thinking about. I'm thinking of the membrane potential. You're right, it's more positive. But when would calcium stop? What membrane potential would calcium stop moving at if we let it move? Was there a hand up over here to give it a go? Yeah? Positive 120, yeah, it was wicked high, right? So we are going to move, if we make the membrane potential instantaneously permeable to calcium, right? If we calculated the E ion for it, it would be positive 150, 160, 140. It's really wicked high, right? Which means calcium will make the membrane potential 160 and then stop moving, right? Is essentially what that is. So we could then predict, indeed, if we were to hit threshold for voltage-gated calcium channel, the membrane potential will go up, and it will head up towards 140, 150, 160. It didn't do that because we didn't leave it open long enough, right? We opened it, and then it became inactive. It's very similar to that voltage-gated sodium channel you've seen before. 3 states: open, inactive, and closed. Okay? So it's headed towards that E ion, but it's not going to make it because we're not going to leave it open long enough. If we left it open longer, it would head there. Okay? So we open voltage-gated calcium channels. We depolarize, we get calcium rushing in, depolarize, and then we inactivate. Those voltage-gated calcium channels become inactive. So what's special about the inactive state? Why didn't we say closed? Does anybody remember what's special about an inactive state? Yeah. See, it won't let anything in no matter what, but why is that different than closed? So close. So can you add to that? Perfect, right? So closed, you can open it. Perfect. Inactive, you can't open it. You can't open it yet. So you've got this state where it was open, so we had flux. It's gone inactive, we can't open it again. So it's gone into a state where we can't get it open. It's got to then change its shape again to go to some sort of shape that allows it to be then closed so it can be reopened. Okay? So we've got a voltage-gated calcium channel that goes inactive, so nothing's fluxing through it, but we can't reopen it, all right? Then as we depolarize, at some point we hit the threshold for a voltage-gated potassium channel. Okay, we get potassium flux out. Oh, we opened it. We get potassium flux out, movement of a positive ion out. It's working towards its own E ion of like minus 100 or something. Okay, so it's going to stop moving when the membrane potential becomes minus 100. So if we open voltage-gated potassium channels here, we get potassium fluxing out. The membrane potential goes more negative, and we are headed towards minus 106 or whatever it happens to be. We're not going to get there because we're not going to leave it open long enough. So someplace down here, potassium channels. I better put voltage gated potassium channels close. Okay? So if we think about this over time, we need to add that element on here. So if we think about time, this is happening slower than the action potentials you've seen before. So this is going to happen over hundreds of milliseconds, whereas the action potentials that you've been looking at before in skeletal muscle and in the nerve happened over a millisecond. Okay, so this is now happening over a much longer period of time. Okay, so if we start the action potential around here, and we'll say maybe that was time 0. They close at around 300. We definitely get, it's 200. And then I'm going to take it out to about 200, 300, 400, to about 500. Okay? So longer action potential than we've seen before. So open a voltage-gated calcium channel, inactivate it. Open a voltage-gated potassium channel, close it. We have periods here. So I guess a result of having an inactive state in a channel, just like nerves and muscles, was that now we have refractory periods, right? Do you remember those? We have periods where this piece of tissue can't be stimulated again, right? It was this piece of tissue, this action potential is highly dependent on those voltage-gated calcium channels being closed. So we can open them. So if they're open or inactive, we can't generate another action potential. Okay, so there is going to be the same phenomenon like we've seen in other action potentials, that you have a refractory period, and we called this the absolute refractory period before. Here we're going to use slightly different language. It's going to take us a little while to get there, but I'll explain. I'm going to put the word down, but we'll explain it. It's called an effective refractory period. So it's like the absolute refractory period, but there's a little bit of a difference. Okay, so here is a tissue being designed so that it can't be stimulated at a time where it doesn't want to be stimulated. Okay, that was what refractory periods are all about, making sure that a cell cannot be stimulated again before it's ready. Okay, this is going to be really critical in terms of making a pump. You can't have the pump pumping and in the middle of a pump cycle try to get it to pump again. Okay, you got to fill, pump, fill, pump, fill, pump. You can't just be randomly throwing action potentials at this thing. So it's pretty guarded in terms of the time of period that it cannot be restimulated. Okay, so the effective refractory period is certainly a 200 millisecond period where you can't do it again. Yeah. It's a sodium. Yeah, so is this leak supposed to be sodium or calcium? It is indeed a sodium channel. Okay? Just like, so sodium, so let's think about what sodium would do. If you made this cell instantaneously permeable to sodium, we're not doing that, But let's say we made the cell instantaneously permeable to sodium, what would happen? So let's think back. Where is the concentration? What is its E ion? What's the E ion for sodium? Anybody remember? Is it a positive or negative? It's negative. Everybody think it's negative? Oh, we got positive over here. Oh, physiology fight. It's awesome. It's positive, okay? It's about positive 61. Okay? So if you make the cell instantaneously permeable to sodium, you are going to depolarize, right? You can also make it instantaneously permeable to calcium, and it'll depolarize. So we have lots of tools to get a depolarization to happen. Okay? Have you read somewhere that it's calcium? Is that where the question is coming from? Oh, okay. So it is indeed a slow leak of sodium, right? So we're not making it instantaneously permeable. We've just got a couple of channels in there, and we're getting this slow depolarization. We hit threshold for a voltage-gated calcium channel. Now we've made it instantaneously permeable to calcium. We get this monster increase in calcium flux and this huge change in membrane potential, depolarize. And at that up around that peak, these are usually timed events. The voltage-gated calcium channel moves from its open state to its inactive state. Okay, so no more flux. So we've really abandoned sodium ever since here, right? This is a product of voltage-gated calcium, for sure. And then we repolarize to voltage-gated, with voltage-gated potassium. And then we're going to start the whole system all over again, okay? So the gates going inactive, is it spontaneous? So I'm going to say yes. But it's not a surprise spontaneous. It is, like when you have we're talking about a protein here, right? That's what these channels are. So even your voltage-gated sodium channel that you learned about before, it was just a protein, right? So it's this protein that is sitting in some sort of conformation, right? And so this is kind of how I visualize these states happening. It's like this, it's sitting in a conformation at rest, and it's closed, right? You hit threshold, right? So you change its membrane potential around it, and it changes its shape, right, to something that will open. But that's really just a transient state, because it's really just changing to this, and now it's inactive. And then we wait, and then it'll click back to closed, right? So it's almost like these states aren't like three absolutely separate states that this thing is going into. It's like the open state is kind of a transition into the inactive, and then you wait a period of time and it goes back closed. So I would argue I can't argue against the word spontaneous, but I would prefer the word timed, that there is just a certain amount of time it's going to be open, there's a certain amount of time it's going to become inactive, and then there's going to be a certain amount of time before it clicks back to the shape where it's closed. Does that help? Yeah, okay, good. Any other, yeah? So does calcium go inactive as you hit? So what I've put on there is a little bit contrived, but the voltage-gated potassium channels have their own threshold. Okay, so voltage-gated potassium channels have their own threshold. As we moved the membrane potential from -60 or -50 up to 0, we hit threshold for potassium. Yes, let's go there. Yes, it does. Yeah, you're absolutely right. So you've hit on it, because I was just going to glaze on by it, hoping that no one would notice, right? Just look away, don't make eye contact when you talk about sodium leak. Indeed, you can't just have sodium just randomly leaking, because eventually you'll just leak all the way up to +61, right? You do have to do something about the sodium leak. And there is, yeah, you will It's complicated. And I don't necessarily want to go into that level of detail, but do just know that we do have to do something about that sodium leak as we go through our action potential. So thanks for messing me up. Yeah. All are following their concentration gradient. There is no pump here, right? In order to restore the concentration gradient, you do need a pump, right? Yeah, so once calcium, and we haven't put them up here, you learned about them already in the first physiology course, right? That you'll have all this flux happening, and that eventually you're switching the spaces that ions are in, and you do then have to have pumps to put them back. The key to this, though, and the key to running an action potential on anything, is that the number of ions that are actually moving to change the membrane potential are really small. So you can, let's say on a nerve, let's say you're on a nerve now and you've got voltage-gated sodium channels opening and sodium rushing in, okay? You can run that action potential for hours and never have a pump to have to restore the concentration gradient, just because so few ions are moving to actually make this happen. So you do, over time, need to restore the concentration gradient, but it doesn't have to be an immediate per beat thing that's happening, okay? All right. Yeah, so do you remember we had those leak pores? Do you remember we set them up right in the very beginning? Like, why is the membrane potential what it is at all? So that membranes have certain permeability, so they have no permeability to let's just talk about a membrane, no permeability to calcium. They have a lot of permeability to chloride, right? Remember, we always took chloride out of the equation. It's usually permeable. Some permeability to potassium and very little permeability to sodium, right? So a membrane all on its own is sitting there with what they call pores or leak channels, right, that just allow these ions to move. And so it just so happens that this cell has more sodium ones. They're a little different, but there are just more sodium ones sitting here, right? So we've constructed the cell a little bit differently. We, like I had anything to do with it, I didn't. The cell was constructed just a little bit differently, okay? So it is not voltage gated, it's just one of those pores sitting in there, okay? So this membrane is slightly leakier to sodium. Every time you got a leak channel, you have to have pumps somewhere, and we always have the ever-present sodium-potassium pump. Yeah, so these channels can be closed, but I think under normal circumstances, under normal physiological circumstances, I would argue no. If the cell wants if the cell doesn't want to be permeable anymore, it's got to pluck it out. All right, if the cell wants, if this cell, if you want a stable membrane potential on this cell, all you have to do is pluck those out of the membrane. Right, that's how a cell would do it, and it was no longer leaky, or it's no longer leaky to sodium. Okay, it's got to take it out. There's really not a whole lot of control over them. Okay? All right. What was I talking about? An effective refractory period. Oh my gosh. So here, because voltage-gated calcium channels are either open or inactive, we cannot generate another action potential here, and that's a good thing. So we can't generate another action potential within the first 200 milliseconds of generating the first one. Okay, that's good for our pump function. There will be a period that's akin to a relative refractory period. Do you remember that relative refractory period where we could generate another action potential if the stimulus was greater than the original? Remember that? So I'm going to write that down, but just note that it is kind of ridiculous for this piece of tissue because it's generating the action potential in the first place. So there can't be a greater stimulus than the one it's generating. Okay, so I'm going to write it down because physics lives, okay? But it's not often applicable to this piece of tissue because this piece of tissue is the thing that generates the stimulus in the first place. Okay, so there is a relative refractory period, so we can't, I'm going to continue writing down here. We can't generate another action potential. Oh, no, we can. We can generate another action potential. And in fact, this refractory period, this relative refractory period, is part of a longer period where we can't generate another action potential that looks like this. Okay, so I would argue from, including this relative refractory period, from here all the way through to about 500 milliseconds since the start of the first action potential is this period called, or period of, post-repolarization. Refractoriness. Okay, what does this mean? This means that experimentally, we can go in, so don't try this at home, but you can take muscle or you can take the SA node out and experimentally look at this action potential and measure the change in membrane potential and then give it a really good jolt, give it a really high stimulus. So depolarize it on your own externally in this period and you won't get another one of these. You'll get something funky, but you're not going to get one of those. Okay, so what I mean by that is if let's say I have my regular action potential, right? And then I'm going to wait till I hit threshold again and I'm going to get another one. All right, so I've got two action potentials here in series with each other, right? That if I tried to generate an action potential early, so this is experimental, so I throw in a little bit of an electric jolt right here. Right, so let's say I try to throw in an action potential experimentally. I'm going to get a little something, but it's going to look weird. There's a little bit of depolarization, but it's not going to look normal. Right, and then I can, if I wait a little bit more, I can try it again in that kind of up to 500 millisecond window, and I'm going to get something that looks a bit weird. Okay, just hang on a sec. And then if I try it again, I'm going to get something that looks just a little bit weird. Right? Only until very close to threshold am I going to get another one that looks the same. And the hypothesis here is we are waiting for voltage-gated calcium channels to close. Okay, so nowhere up here have I put voltage-gated calcium channels closing. It's because they don't close at a point. They seem to lazily close over a region of time. And we get enough closed by about that 500 millisecond point that then if we hit threshold now, we get another action potential the same as the first one. Okay? But if a stimulus comes in before then, we're not going to get an action potential because we don't have enough voltage-gated calcium channels closed. Yep. Of the other one, yeah. So there's another one here. That's right. So that slow sodium leak is happening during the post-repolarization refractoriness of the action potential prior to it. Yeah. Okay, so essentially the hypothesis behind why we can't generate another action potential for this longer period is that we're waiting for those voltage-gated calcium channels to close. So if we were to put an indication on this action potential as to where voltage-gated calcium channels close, it's going to be over this period. Okay, so we're not actually going to put an arrow to a small region as to where this activity happens. We're going to put a line to a large period of time. 300 milliseconds, we're waiting for these things to close so we can reopen them. Okay? But this is good. This obviously is going to be required in order to get this thing to pump and pump effectively. Okay? So we will have full excitability at the end of the post-repolarization refractoriness period, and bam, once we hit threshold, we're going to get another one just like the first one, all or nothing. Okay? So life is good. Okay, so let's put this cell into the heart and see why we care. All right? So let's pop just back and do another look at the heart here. So if we're going to look at this electrical system, okay, the electrical system is the trigger for contraction, right? Okay, so the trigger for contraction of this whole pump is going to be that action potential we generated. Okay, so the trigger for contraction is that action potential, and then we're going to generate that action potential just like we did spontaneously. Okay, so the action potential starts in those specialized cells, the nodal cells. And for particular reasons that we're going to get into in a moment, it starts specifically at the sinoatrial node. So we said we had SA node and AV nodal cells. The start SA nodal cells were up in that right atria. The atrioventricular nodal cells, okay, at the junction between the atria and the ventricles. Okay. So essentially what's going to happen is that sinoatrial node is going to spontaneously depolarize, just like we talked about. Right, so this is our spontaneous depolarizer. Okay. And it is going to spontaneously depolarize, so that cell itself now has changed its membrane potential, and it is electrically connected. Right, so it's got gap junctions. It's electrically connected to atrial myocytes, so muscle cells of the atria. So it's going to take it. So its change in membrane potential will be immediately translated into a change in membrane potential in the atrial myocyte. We change the membrane potential of an atrial myocyte, and we're actually going to get contraction to occur. Okay, we'll talk about that in a minute. We're going to get contraction to occur. Those atrial myocytes, right, so that's this. We got this spread, wicked fast spread of that change in membrane potential to both atria, the right and the left atria. So the right and left atria are connected. The action potential is started and spread to both of them. This is why a right and a left atria of a right and a left heart contract at the same time, right? We gave it the same signal. We gave it the same go signal. So we get the right and the left atria myocytes contracting. Those atrial cells are electrically connected, so through gap junctions, to the nodal cells, AV nodal cells. So this AV nodal cell will now change its membrane potential. All right? It then passes, that nodal cell has a similar membrane potential to the one we just looked at. It then is electrically connected, so through gap junctions, electrically connected to other tissues that are excitable. Okay, so they have fancy names, but essentially all they are cells that can change their membrane potential. So the bundle of His. So the AV node is directly connected electrically to the bundle of His, or cells sitting there. The bundle of His is directly connected to different bundle branches, so now we can conduct an action potential down to the bottom of the heart. Okay? The bundle branches are directly electrically connected to Purkinje fibers. Okay, so these Purkinje fibers are now going to go from the bottom and head up, and those Purkinje fibers are then directly connected to myocytes of the left and the right ventricle. So now we're back to exciting a myocyte, right? And so because the electrical signal came down and then split and then went up the right and left ventricles, then the right and left ventricles contract together because they had the same input. Okay, so that's why a right and a left heart, two pumps, contract as one. They had the same input. Okay, they had the same stimulus input. Okay, this spontaneous depolarizer, the sinoatrial node, spontaneously depolarizes 70 to 80 times per minute. Okay, so we have this depolarizer spontaneously depolarizing 70 to 80 times per minute. Okay, these are our pacemaker cells. This is why your resting heart rate is about 70 to 80 times per minute. You can thank that to the sodium leak, right? Resting heart rate all comes down to that funny sodium leak, right? So it's sitting, resting, depolarizing 70 to 80 times, and it's sending its input out, so the heart beats 70 to 80 times per minute. The atrioventricular node, if you were to take it out and watch it all on its own, I'll try it at home, but if you were to take that out and look at it all on its own, it has its own spontaneously depolarizing rate. It happens to be 40 to 60 times per minute. Purkinje fibers are also spontaneous depolarizers. So if you were to just look at them independently and their leak rate, they would spontaneously depolarize 15 to 40 times per minute. So indeed, if you had the heart working normally, it would contract at 70 to 80 times per minute. But then why doesn't this rate get in the way of this rate? Like, how is it that we can, how possibly can we have three spontaneous depolarizers and only go at the rate that one is dictating? Is the strongest. Okay, good. I'm going to question the word strong, though. Action potentials are really just all or nothing, right? So we can't have a bigger one or a smaller one. Yeah. Exactly. That's the brilliance of the refractory periods. Okay, we are in refractory periods, and then at the end of the day, it's just the one that goes the fastest, right? The one that goes the fastest is going to drive the rate. So for instance, if you had no sinoatrial node, let's say you had a heart attack, and you had inhibited blood flow at the sinoatrial node and you destroyed it, what rate would your heart beat at? You see it? What rate would your heart beat at? The next 40 to 60, the next fastest one. And if your SA node and your AV node were wiped out, what rate would your heart beat at? Purkinje fibers. Okay, have a good weekend.